will start.
Yep. Well, good evening, afternoon. Welcome. Please find yourself a seat as we're ready to get started. It's my pleasure to welcome you to this thematic session that we've entitled Clovers Around the World. I dreamed this session up about a year or so ago, and I contacted Derek Woodfield to be my co-organizer, and so the two of us are delighted that you're here for the session. Not only are we going to talk about clovers, particularly at the beginning of my paper, I'm going to discuss the life and career a little bit of Dr. Norman Taylor that some of you need to be enlightened about a little bit. So again, thank you for being here, and now Derek is going to take over.
I am. So I just want to introduce the first speaker in this session, which is the revered Professor Emeritus Ken Quisenberry. Ken had a 35-year career as a professor in forage breeding at the University of Florida. He has since then invested another 12 years as an emeritus professor cooperating on turfgrass and forage breeding there. Interestingly, and for this symposium or workshop, he did his PhD with Dr. Taylor at UK and co-authored the book on red clover with Norm Taylor. He's been a developer of 24 forage and turfgrass varieties. So Ken, it's all yours to talk about. Norm first.
If I can get rid of this little icon and get this to go full screen, we'll be in good shape. There we go. All right. Well, again, thank you for being here and being interested in our symposium today. Like I said, I'm going to start. Now, will this advance? Okay, here we go. All right, so I want to start by talking a bit about the life and times of Norman Taylor. Dr. Taylor was born near here in Bracken County, north central Kentucky. Was in the Merchant Marines during World War II. Got his undergraduate and master's degree at the University of Kentucky, went on to Cornell, came back and progressed through the academic ranks from 1953 to retirement, like some of the rest of us, in 2001, continued to work as an emeritus professor for another 9 years until his untimely passing in 2010. That's a little blurry picture, but I was the best one I could find of myself, Dr. Dick Smith in the middle, and Norman, who I call the three clover amigos at a time, and Ray Smith particularly will recognize some of those. I had the great fortune of obtaining my PhD with Norman at Lexington in 1975, and unlike many people, have had the good fortune of continuing to work in the area very similar to where I did my PhD, and then worked and cooperated with Norman and with Dick Smith over the years. Ray Smith, I saw you walk in. Recognize that picture? Taken at Overton, Texas, I think Trifolium Conference, where we went out to a little graveyard and collected buffalo clover, I believe. That's what we're looking at there. Trifolium reflexum. And one of the points I want to make about Norman is he had a passion for collecting and assembling species of the genus Trifolium. More so than any person that I've known, and did a tremendous job of that throughout his entire career. I put in this slide second generation legacy, a picture of me in a field of one of my red clover cultivars in the Pacific Northwest. It's really third generation because Norm's master student, Dr. Elmer Gray, is really the individual who mentored me and encouraged me to go to graduate school. And Elmer was professor at Western Kentucky, where I got my undergraduate degree. So I'm really almost third generation. But in developing this talk, one of the things that became apparent to me was that I'm not sure we're pulling in the next generation of young scientists who have the passion and the love for the crops that perhaps people like Norman did. Norman was a prolific writer. He was also prolific in germplasm collections. This is a list of what I could pull together of funded and organized germplasm collection trips. But what you don't see on here is every trip that Norman made outside of Kentucky and several inside Kentucky. Like some of my kids would say, Dad, what are you stopping for? He was stopping to look at clovers and see if there's one that he needed to collect along the way. And you'll see Brian Irish pulled this together for me, a little over 800 accessions in the Grin collection that attributed to Norman. Major publications, signal publications really, the first report of interspecific hybridization in red clover, Norman and I, some work we did while I was a PhD student, really not part of my dissertation, on doubling chromosome numbers. Then he did the Tri-Society's Clover Science and Technology book and cooperated with me in the Red Clover Science. If you haven't seen the Iowa State publication with Gillett and Taylor on the world of clovers, that's something I would strongly recommend you get your hands on. Then involved with Ellison and others on the molecular phylogenetic study, and finally Norman's summary paper of his career published in Crop Science. So I hope that gives you a little background on Dr. Taylor and his contributions to the world of clovers. I'm going to switch now and start talking about the genus and try to convince you why every one of you should be a clover lover. So the genus consists of more or less 250 species, but only somewhere around 15, 16, maybe up to 20 have at all been cultivated in agriculture, but from one who has done some germplasm collecting. There are many, many others that in native rangelands and native sites form an important part of ecosystems for grazing animals. The genus has been evaluated scientifically for well over 100 years. There are 3 primary taxonomic reviews that I've listed there, and I'm going to be using the sections of the genus proposed by Ellison and others for what I'll be talking about in this presentation. Now, I don't expect you to read the names across the top up there or even to be worried too much about the numbers, but just to point out that depending on who you talk to and who decides whether to split or combine a species or call it a subspecies, there are around 250 species in the genus. The first column over here that's called Chronosimium, Ellison and others made that a subgenus of the genus Trifolium, and those are the hop clovers, the yellow-flowered primarily hop clovers in Chronosimium. And then we'll talk about species in some of these other sections of the genus as we move through here. This is another reason. If you're a molecular biologist or if you're interested in ploidy level, this is a genus you should be working with. Look at the range in chromosome numbers in the genus Trifolium. Base numbers of 5, 6, 7, and 8. I know of no other genus that has that range in chromosome variability, and then multiple ploidy levels on out. And, you know, some young scientist who just needs a couple of publications, get some seed and grow some of these and publish chromosome number. There's still 50 species that we don't have published chromosome numbers on. Spread around the world, I lumped Eurasia. There are 141 in Eurasia because it's kind of hard to define where Europe and Asia kind of separate. So there are those, 41 in Africa, Western North America, a little over 50, 7 in Eastern North America, and that's my group of the others. Eastern North America, interesting. Only 7 species. There are other states of which Eastern North American species have been collected. These are the ones that are in the Grin system that are shown here. But there's certainly collections from Texas, Louisiana, Arkansas, Missouri, South Carolina, I'm sure. I would just point out the one little green arrow right there Do you know where that is, Derek? That's Ken's front yard in Gainesville, Florida. There is an accession in the Grand System that says 6828 Northwest 39th Avenue. My front yard. That's Trifolium carolinianum. Okay, so, Western United States, California, Oregon, and Washington are the predominant locations of species, but scattered up through the Rockies, not much goes past the rain shadow of the Rockies, though. Central and South American species, Chile, probably the predominant country, followed by Mexico, but a scattering in a number of other South American countries. I'm going to switch now, and really, I have no data for you. Okay, this is the picture show to convince you of the amazing diversity in the genus. Okay, hold up your hand if you know rabbit's foot clover, Trifolium arvense. Isn't that a great-looking species when it gets that fuzzy head on it? Okay, a base number 7 tetraploid out-of-section Trifolium, distributed Old World, New World. Strawberry clover. Okay, Trifolium frugifera, base number 8, widespread section vesicastrum. Look at those, how those heads become inflated with the calyx in them. One of the most unusual ones that I know of, Trifolium pauciflorum. It looks like a dandelion puffball almost. Base number 8 and the section trichocephalum. Subclover? Yeah. Pretty unusual seed head morphology, right? Essentially, the calyx teeth become soil diggers and kind of pull the pods into the soil as it dries. Very unique. Base number 8, again, section Trifolium. One of the best pictures that I have, and one of the most unique species in the genus, as far as I'm concerned, is Trifolium polymorphum. So in case you're not seeing that, these are peanuts. These are single seed pods developed from a single floret in the axis of the leaf and stem on a structure that very much resembles a peanut peg and bears itself into the soil. And these are typical aerial flowers that look like a pink flowered white clover. We have shown with some cooperative research for some folks in Uruguay, these are primarily cross-pollinated, and these are exclusively self-pollinated. So it's a darn conflicted species. It doesn't know what to do. So is it heterozygous or homozygous? Who knows? But it's a fun species, thanks to former graduate student Carlos Acuna from Corrientes, Argentina, for that picture. Trifolium jokerstii, newly described about 10, 15 years ago from Butte County, California. The only bright yellow flowered species in North America, in Volucaria. Unknown chromosomes haven't even been counted on that one. Trifolium aminum, again, unknown chromosome number, thought to be extinct. Beautiful. Aren't those nice looking heads on aminum? Rare and endangered, but we've got some collections in the Grand System now. A blue flowered, Trifolium alkali, section vesicastrum, very blue-purple flowers. So that shows you some of the range in the genus. Yeah, there's my friend who likes the North American species. This is buffalo clover, Trifolium reflexum. It has white, these pink flowers, the more dark red with the white standard petal, and then solid deep burgundy flowers. Norm made a number of collections of buffalo clover around Kentucky. We're going to look at some of the ploidy variability. This is rose clover. N equals 5, diploid, section Trifolium. Has been investigated by folks like Ray Smith and others in the U.S. Trifolium stellatum. Look at the calyx teeth on that species. Really unique. This is one of those N equals 6 species in section Trifolium. You've seen pictures of this. Jimmy Henning posted a great picture on Facebook yesterday of crimson clover. Nothing prettier than a field of crimson clover in the deep south about a month ago. It's mostly in seed now.
5 minutes, Ken.
Okay. Trifolium rubens. I actually did some crosses with this when I was a student, called by various names. Red feather clover. There's some folks marketing seed for home garden uses for ornamentals on this one. A diploid N equals 16, or N equals 8, 2 N equals 16. Trifolium macrocephalum. I have not seen this one personally in flower. Big heads, huge heads. We collected some seed off the ground up on the Columbia River in Washington, just across the river from Oregon. The seed had already matured and fallen on the ground. It's an involucraria. Trifolium canescens, a hexaploid. So just look how we're going up in chromosome numbers here. In section Trifolium. Zigzag clover, I made some crosses on this one in my PhD work. Trifolium medium. We showed in that work that you can have 64, 72s, and 80s, and some odd ones in between there. Highly rhizomatous, strongly perennial. We've collected species of that. I think Ray Smith and I collected some of those in southern Bulgaria. Medium. And then here we go, Trifolium pannonicum. I'm not sure that we really know. I'm not sure everybody counted all those 128 chromosomes, but section Trifolium, it's a multiploid somewhere in the 16 ploid range. Beautiful species. So that finishes the picture show. I hope this discussion and the photos I presented have demonstrated for you the amazing diversity in the genus Trifolium. As I said, as we mature, we begin to think about the next generation of scientists that will continue and enhance our work. And hopefully some of my comments and those that follow will lead to a resurgence of interest in this wonderful genus of plants. And we'll be able to continue the legacy of Dr. Taylor. The other major topics that follow will be introduced, but here's a list of them. And I'll leave you with this thought. If you're not aware, Cicely Mary Barker published the Flower Fairies of the World, and there's the red clover fairy. And so clovers are important, and that's not a fairy tale. And maybe we have a minute or two for questions.
Thank you very much, Ken. We do have time for a couple of questions, but while we're doing that, Randy, if you want to switch presentations, that would be helpful. Is there a question? I'll give you a comment then. When one of my first bosses, when personalised license plates for cars came in, got the license plate called Clover. And one day he couldn't work out why he was driving around town and everyone was honking at him. And somebody had gone and covered up the C, so he was driving around with a car with Lover on.
I do have a Gator vanity plate that has the UF logo, but you can only get 5 characters if you put the logo. So it is CLOVR. All right. So our next speaker is Randy Dinkins. Randy is USDA ARS research geneticist at the University of Kentucky. Works with molecular genetics, molecular biology. Started out with Arabidopsis, I believe, and then these days focuses on tall fescue and red clover. Yes. And he tells me that he lived down the hall for Norman for part of his career, and part of the reason he works on red clover is Norman kind of suckered him into it. So.
And that was exactly where I was going to start off. When I came on to the USDA ARS in 2004, Norm kind of corralled me and wanted me to work on red clover. And so that was part of my job. So when I first put in for an abstract here at the Congress, I was going to do that first part, molecular approaches in analysis of red clover and rhizobium symbiosis. And then Ken pulled me in and said, why don't you do some more stuff on showing what the molecular biology or where it's at right now with red clover. So there's part 2. And this is where Norm, I think, would want to see where things were at. When I started off with him in 2004, again, he had retired, but he was still going strong. And so we were out in the field doing some work on some of the non-browning clovers. And Dr. Kagan picked that up, and we published some of that work a while back. And he was also going out there and spraying some plants that he had done some selections and crosses from 2,4-D selections from Florida into the Kenland variety. And I'll talk about a little bit at the end where we're at with that. But as an Arabidopsis person, I wanted something that could self. And red clover does not self. So we grew a bunch of these different clovers in the greenhouse, and some of these did self. Some of these are self-pollinating, but red clover is not. So for molecular biologists looking on an outcrossing, highly heterozygous, heterogeneous population, it's difficult to do molecular biology. But we did pull up and do a transcriptome in red clover early on. But since Ken asked me to talk about where things are at, things have really progressed, and we are now with a number of different assemblies that we can use to look at molecular biology. 2014, the first one was published. Then in 2015, another one from group from UK and Wales and Ireland came together, and more recently from ARS, Madison. And there are a couple more from different places in China, but we don't have the annotation for those, so I don't have them listed here. But this is where things have come along. So the first clover assembly from the group from Czechoslovakia, from this cultivar Tatra, they had a large number of different, basically, contigs that you could map your genes onto. The one from the TGAC had 39,000 contigs, but at least it had 7 chromosomes. So now we could actually map genes. And the more recent one is from ARS Madison, where we have basically 7 chromosomes and a few loose contigs still out there. And this one's on NCBI. So here's a depiction where you can see the genes on chromosomes. Here's a list of all the genes that are on there and some of the reads that are in NCBI where you can actually see what the genes, how they're being expressed. And so here's the 7 chromosomes and the assembly from the red clover, from the ARS and the TGAC. And they're very similar, but there's some differences, and some of the loose contigs have come into focus. So now we have 7 chromosomes and a few loose contigs out there. Oops, did go too far. So here's looking at the red clover assembly. This is the one I just showed you, TGAC and ARS. At the 100 kb level, synteny. And this is a synteny with the different clovers and Medicago truncatula. So N equals 8, but there's still very little with Medicago, but a little bit more with subterranean clover, N equals 8, western clover, N equals 8, and of course white clover, N equals 8, but at 2N equals 16. So here's the assemblies, and here's where they match on the other chromosomes for the different species. So this is the ARS assembly with white clover at 60 kilobase contigs synteny. So it's fairly good to the clovers. Medicago, truncatula, again, closely related, but further apart. So I'm interested in looking at gene expression and mapping these genes. Again, this is the NCBI. And I'll talk a little bit about, since I'm talking about the rhizobium. So here's just a little section on chromosome, I'm not even sure, 7, I think. And here's just a regular gene. and this showing what the expression of this particular gene is in just different tissues. So this one here is actually what we've called nod minus, because there's non-nodulating, and this one is nodulating. So it's basically the same for this particular gene, which is a cellulose synthase gene. In the nod plus, we have some of these what's called nodule-specific cysteine-rich peptides that are only expressed under nodulating conditions. And so in the N minus, nod minus samples, you get no expression and very high expression in the nod plus. So this is one of the things I'm interested in, and I'll talk a little bit more about that in a little bit. So if we're looking at gene expression, we look at the long reads, iso-seq. So this goes through and shows that when you do the iso sequencing, it goes all the way through from the poly-A site all the way to the 5' end. But most of the sequencing that I've been doing is short read sequencing, or RNA-seq, which is about 150 bases. And so this shows the gene structure. So this is an exon, and the dotted lines are introns. So this is the expression of this particular gene, showing the different profiles. And using the long read, you can actually do looking at alternative splicing, which is what may be interesting for different genes. And it shows that we have alternative splicing here that you can pick up when you do short read sequencing, but not as well. And this is what the long read sequencing is good for. But for the thing with red clover, again, it's a highly heterogeneous species, heterozygous because it's outcrossing. This is one clone. So you see significant SNP or single nucleotide polymorphisms in this one clone compared to another clone. And in the field, it's going to be, this is, again, one genotype, what it originally from. And so you see the significant variation already in this one gene. I'll talk about this gene here a little bit later. And so the other technique in molecular biology that we're using is looking at particular genes. Again, in Arabidopsis, we are focusing on particular genes. So one of the genes, or the genes that since you have so many polymorphisms, it's kind of hard to nail down if you have a mutation, is it effect or not? So we're using what's called CRISPR-Cas mutagenesis, transformation protocol that comes in and basically knocks out a piece of the chromosome, the chromosome, and so that disrupts the gene. You can specifically target sequences that you want to knock out. So this is why you need the assembly or the genes that you're interested in looking at, because you need to construct a target sequence to knock that gene out. And we use regular tissue culture. Again, Norm was instrumental in getting all this tissue culture up for red clover. And this is what we're using from Mike Sullivan's laboratory and some clones that, again, Dr. Quisenberry provided to us. So one of the first genes that I looked at is since we're interested in some of the isoflavones, biocanin A, formaminonin in our laboratory, one of the genes that's in the pathway, isoflavone synthase. So it's from the phenylpropanoid pathway. And here is the progression from phenylalanine to biocanin A and formaminonin. What happens if you knock that gene there out? What do you have? So we did. We call it a knockdown because we deleted 9 base pairs. So there's 3 amino acids missing. It was a 9 base pair. So it's still in frame, so we're calling it a knockdown. And we sequenced, and sure enough, this is the region for the gene, for the ones that were knocked out. But you can still see all the different lines with these genes, the variation in this gene in red clover from one original cross. We had to cross with some other different lines to get it to get homozygous lines out. And so here's a homozygous line for the knockout. but all these lines here are the different SNPs in this isoflavone synthase gene. So again, we sequenced those that were not inoculated and those that did have nodules on there as well. And so this was just some of the verification that we had in terms of the levels of formaminonin, biocanon A. So here's the homozygous wild type, here's the heterozygous line from the same cross, and here's the homozygous mutant. So showing that we did knock down the formaminonin, knock down biocanon A, as well as the intermediate products, genistein and diazin. So the knockout worked. And we're looking at whether or not it nodulated or was this affecting in terms of nodulation. And the answer was no. It's not an isoflavone, but a flavone that's probably in charge of rhizobial inoculation. But using these same lines, we could look at gene expression in the between the nodulating and the non-nodulating, and that's what we came up with. We looked at the RNA-seq, or gene expression. So here's the gene expression of that isoflavone synthase gene. Here is in the NOD minus, a wild type. So here's the gene expression levels, basically, of your wild type. And when we did knock out in the nodulating ones, we saw that the isoflavone synthase gene was repressed. So it's kind of like, hey, there is an effect of this nodulation on the expression levels of this particular gene. Same thing with the upstream genes, PAL, and which is up here, and the chalcone isomerase in the pathway. Again, we see the changes in terms of those that are nodulating versus those that are not. The IFS, the knockout mutants, compared to, so PAL has several different isoforms, and these are the two genes, and here's the chalcone synthase. And there was significant changes in terms of the expression profiles, whether or not it was knockout and whether or not it was from a NOD plus or a NOD minus. There was not much difference between that and that, but it seems like it's under whether it's not nodulating or not. And some of the same was observed in some of the ethylene response factors, some disease resistance genes. So this kind of showed us that knocking down the IFS gene also affected some of the other pathways in reception or perception of bacteria in the soil. So again, just looking at, I'm interested in the nodulation, and in the legumes, as in the galligiloid species, they have an intermediate type of nodulating or not symbiosome or nodules, in that they continue, it's like a meristem, unlike soybean, which is a determinate nodule. So what is the difference? And one of the things that we have found, or others have found, is that there's production of called nodule-specific cysteine-rich peptides. Medicago truncatula has one of the highest number, is about 700 now. And so we sequenced from red clover 450 of those that were not previously in the annotated genome. So these peptides are both antibiotic in the sense they knock down microbial, and also are positive in terms of the microbial population when they are invading and forming the symbiosome. What are these guys for? We don't know. So we looked at gene expression profiles. So this is the number of genes that were significantly different from the nodulating ones versus the non-nodulating ones. And this is from the non-nodulating versus the nodulating ones. And this higher number here is in part because there are so many of these being expressed. So again, I came back to this picture just showing the expression in terms of the NOD minus, not expressed. Here, some of the NCR, that I showed up earlier and expressed under those that are being, where there's nodules. Talking about this particular gene, again, it's not expressed under the NOD minus, and this is expressed under NOD plus situation. It's a transcription factor. So kind of curious, what is this transcription factor doing? We went through the same process using CRISPR-Cas knockouts, and we've generated a number of knockouts in this gene. This is the region that we're knocking out. And so here's some of the different ones using the CRISPR-Cas where we've knocked out. These are what's called hemizygous, most of them, because one allele is knocked out, the other one is still a wild type. Here's our wild type sequence from the parental line that we used. And some of these, again, are in frame, but we have some where it shows the stars because it's knocked out of frame, and now we have a dead enzyme. And this is what we're looking for. What does this gene do in terms of the nodulation? Going back to this again, so here's our region that we knocked out or we're targeting. Again, so here's some of the polymorphism that's in this gene. If there is a missense mutation in this gene, but we don't know what this gene is. To do molecular work in red clover, this is one way to attack it, is to do knockouts. In terms of nodulation, I went forward, didn't I? And tried to find out what these genes do. Coming back to the 2,4-D, originally I talked about 2,4-D. We're also interested in looking at gene expression profiles of the 2,4-D mutants or 2,4-D selections that Dr. Taylor did in the field. So we're carrying this out. This is a project with Lucas Araújo, a PhD student, Michael Barrett. And so these were sprayed with 2,4-D, looking for resistance or tolerance in these fields. So these were sprayed at 2.2 kilograms, which is basically 2 times the 1x rate, and they're surviving. And so here's Kenlin, the control, not doing so well, but they still, after it doesn't knock out Kenlin, here's injury and regrowth after harvest, whereas the UK line is doing better. It's not better than the Kenlin, but still there's room for improvement. So what we're doing is looking at gene expression profiles. So the group from Florida did some SNP mapping, and on the 7 chromosomes, identified locations on the chromosome where they're associated with this tolerance. So we're going through and looking at gene expression profiles, looking for if there's changes in these regions or if we can find a knockout. Since this is not really it's not a qualitative trait, but a quantitative trait, it's doubtful that we'll find a gene that's going to knock this stuff out. But hopefully we can find, this is the last slide, hopefully we can find some expression profile differences or at least some mutations in genes in this region that will help us identify what we're looking for to get some tolerance for 2,4-D and red clover that Dr. Taylor has started there in Kentucky. So thanks to the people that work in the lab and as well as some of the other labs, and of course, Dr. Taylor. Thank you.
We have a couple of minutes.
That was a
Couple of
fly.
minutes for questions. Anyone question for Randy? It looks like you convinced them. Any questions? Give Randy another round of applause, please. Thank you.
Oh, you have the
The
did I not get it up here? Try that again.
So the next speaker in this session is Dr. Heathcliff Ryday. He's a forage legume breeder at the USDA Forage Research Centre in Madison. He breeds red clover, cover crops, alfalfa and minor forage legumes. He's been a breeder for over 20 years and is also the research leader for the Dairy Forage Research Unit, which I think puts you squarely in the current generation of breeders.
Whatever that means.
All yours.
Okay, thank you. I actually appreciated that Randy talked first. So he was supposed to cover the molecular. I'm going to talk more about kind of practical breeding and kind of where we've been and kind of where we're going or what's happening out. As a breeder, I think I spend most probably a third to half my time thinking about seed production. So that once you get into making varieties, you realize that seed production consumes a lot of your life. And so this is kind of a seed production graph, U.S. seed production graph for about the past 100 years. As you can see, something happened in the 60s where there was a drastic decline in red clover acreage. But the key is, I guess kind of in my lifetime, the key is a red clover seed production has stabilized. And so the assumption is that whatever the red clover acreage has also stabilized. And in the U.S., it's about 5 million kilograms per year of seed produced. Now, looking globally, about half the seed production in the world is occurring in the U.S., and then most probably the other half is occurring in Europe. The green line is the European Union, and then I'll talk about my next slide. Oregon dominates seed production in the U.S., and so that's kind of this orange line. Well, it's a little more detailed. Canada doesn't really publish seed production statistics. They publish usage, so this is from the Ag Census. So Canada is kind of a major player too, and they're also producing about seed. So the total global seed production is about 1 million kilograms of seed produced a year. And then there is some evidence based on the past 20 years or so that seed production is slightly increasing. So for whatever, it does appear that red clover seed production is both in the U.S. and in Europe is increasing. So now a little background, for politicians and other people, like we always like to define the value of something. So there's no red clover acreage statistics globally. So you really have to kind of take the seed production value and translate that into some type of acreage. So I'm listing all my assumptions, and the polite way to say assumptions is I'm modeling. But one assumption is based on the rate you can kind of estimate 4.7 million hectares. If you take the ratio between the amount of alfalfa seed produced and the known alfalfa acreage in the U.S., you can come up with a different rate. And so I split the difference. So my assumption is globally there's about 3.9 million hectares of red clover. And based on the increase that we're seeing in seed production a year, that rate might be increasing at 0.12 million acres a year globally. The retail market value of the seed, it's about $0.1 billion globally. And then assuming the 3.9 million global hectares and assuming 8 megagrams per hectare of forage, we can estimate the forage value globally is about $10 billion in forage production from red clover a year. Making some other assumptions, we can assume that about $0.8 billion of nitrogen value is produced by red clover globally. We can estimate the milk value at approximately $22.9 billion if we translate that red clover production into milk. And if we were to translate it into beef, it's about the same value. Now, you can play around with the numbers and come up with your own. Take your pictures, then you can plug in your own numbers if you don't like mine. But yeah, that's kind of the value of red clover. Seed production kind of globally, if we look a little by country. So in the European Union, these are kind of the top 10 countries producing. France dominates, and then Czech Republic is there. Major changes perhaps. Germany seed production seems to be increasing, and for some reason in Lithuania, recently there's been a jump in red clover seed production. What's interesting in the U.S., starting in about 2000, essentially all seed production has shifted to Oregon. So there's been a mass, like all seed production is now concentrated in Oregon and the U.S. And again, these two sites, this represents most probably about 90% of all global red clover seed production. This is based on U.S. data. This represents historically, for the past 100 years, the number of farms that were growing red clover seed. So you can see this massive decline. I had to kind of blow up this area so you could kind of see. So at this point, there's 200 farms in Oregon producing 35% of red clover global seed. So to me, that's just kind of interesting to think about. And this amount of seed is covering 0.5% of all agricultural land in the U.S. So 200 farms are growing seed that's covering, 0.5% of U.S. Agricultural acreage. Over here, we also have, to me, this speaks to kind of the consolidation that we've really seen, concentration, specialization of seed production that's really happening. Also, the acreage per farm, the hectareage per farm growing has also increased, particularly in Oregon. So now, it used to be five acres, but now they're up to like almost 40 hectares per farm on average. And I have Oregon here in blue, and then all other U.S. states excluding Oregon. So there is still some activity outside of Oregon, but you can see it at a lower level. Now, seed yield increases. There's been this here again is from the U.S. You can see this is red clover seed produced kilograms per hectare. We've seen dramatic increases. So in Oregon, seed production rates are increasing at 9.2 kilograms per hectare per year. And these are pretty decent. They're pretty comparable to alfalfa seed acreages. Outside of Oregon, it's about a third. But even outside of Oregon, it's increasing. And the rate of increase is about 5% increases in seed production yield. So as forage breeder, we always get a bad rap because corn yields are increasing dramatically. But actually, if we look at red clover seed production increases, so corn is increasing at 3.6%. So in some ways, red clover seed production is increasing at a rate faster than corn increases. Now, the strange thing is, no one's really, I don't know if anyone's really breeding for red clover seed increase. There's really not a lot of organized effort on this. And again, a lot of this could be management. It's unclear. But after doing this talk, I need to go talk to Ryan Hayes in Oregon, and most probably we need to maybe look at this a little closer. Okay, so now I'm going to start talking about, that's kind of a background on where we're at. So what are people breeding for globally? The main trait of interest continues to be persistence. In North America, these are kind of the diseases, and I picked the diseases where I know that people actually making varieties were testing for these diseases, or at least looking at them. So everything, I'm focusing on simple traits because these are the ones that I know that people are actually incorporating into breeding programs and trying to make varieties. So these are kind of the ones in North America. Europe, sclerotinas still dominate. There's been a interest in southern anthracnose as potentially emerging disease in Europe. One thing that we've definitely seen in the last 10 to 20 years is a big interest on mixed pastures, grazing. So this here is the mutton clay from Switzerland. This is, if I can read German, clover grass mixtures. So that, they're kind of talking about that. And yeah, here also in the US, when I first arrived, I figured the remaining market was really grazing. And so I had also focused my program really about looking at mixed pastures and getting legumes in there, increasing persistence. And then just shout out to around the world, the New Zealand, they have relish. So they've also been breeding for, again, grazing type red clovers. Another thing that we see in the past 20 years is breeding for specific climates or locations. So there's the first, they claim, I haven't checked, but in Brazil, the first red clover for tropical, so they have this mesclador. Ken Quisenberry already mentioned Barduro, so we have specialized clovers as winter annuals in subtropical climates. This here is the Scandinavians are really interested in breeding, looking at diversity and breeding for their Nordic climates, so there's a whole project on that. This is the UCLEG. This was a project that going on in the European Union. Again, the Europeans like to look at diversity, and they're trying to figure out, using this diversity, can we move our germplasm around and using that information. Again, I mentioned in Switzerland, mutton clay, these are kind of land races that they have, and the Swiss have put a lot of interest into that. Now, I've been sucked into the cover crop world, so the question is, I think there is actually, so I've been talking, for a while there we were really focused on pasture and perenniality, but I think with this interest in cover crops, looking at kind of short-term, high-producing red clover, I have talked to various people. There may be renewed interest in that. We at this point we're talking about doing varieties for this type of variety trialing for this type of situation to see if existing varieties can meet this need. If not, we may start doing a little bit of selection, but that's a potentially emerging area. Okay, breeding for biomass. So there's a continued focus on increasing biomass yield. So we have diploids. In North America, everything's diploids. In Europe, there's a big emphasis on tetraploids. What comes with tetraploids is the emphasis on seed production, because tetraploids have problems with seed production. So these are various breeding sites. So this is kind of Swiss. This is the Swiss breeding station there outside of Zurich. This here is Wisconsin. I got this picture from Serbia, but kind of example, and then this is here a seed breeding site in Norway. Now, breeding progress. So everybody, and actually I didn't have Fernando's picture. You had a nice picture over time. This is, everybody loves these pictures. I was in Hokkaido, Japan a couple years back, and 2000 variety, 1960s variety, it's dead. Everybody likes to show these pictures, and they're great. So I'm supplying some numbers to that. So these are kind of studies where people have looked at selection gains, like how are we doing in terms of selection day. Just so you're aware, this one is per decade, this one's per year. So you kind of have to divide these numbers by 10 for comparison. And so this is kind of a Swiss study. These are diploid, tetraploid red clovers. This is actually from Derek. These are New Zealand numbers, and then there's a bunch of Wisconsin numbers. So what can we learn from these studies? Genetic gains in tetraploid for yield have been greater than diploids. So that's kind of, that's supported by both the Swiss study here and this one out of New Zealand. Genetic gains in persistence have been greater than for biomass yield. So these are selection gains for persistence, and then these are for biomass, and these are actually pretty comparable to the Swiss gains for biomass yield. The other thing we see, gains have been greater for later years. So gains are usually less in first and second year stands for both persistence and yield, and gains tend to have been, they're progressing at a faster rate in later years. Again, these are another visual. So this is kind of the classic. These are 3-year or 4-year red clovers, and their persistence has definitely increased. This is here a classic study that Dick Smith, my predecessor, did, showing again that a lot of the gains have been by extending the longevity of the stand. And then this is a nice thing here I found from Aberystwyth, the Welsh program, and they're again focusing on that 30-year yield and showing that they're making improvements there. So I wanted to actually, these percentages, you can play all types of games with percentages, so I actually wanted to show, figure out, actual ton, megagrams per hectare that were increasing yields. So this here is actually, I have a database of 100 seeded sward trials from mostly America, a couple stray Canadian, maybe New Zealand and French. Other ones, I forgot, I might even have a Polish one in there, but most of them are from the US. And these are essentially total biomass for the trial. And the trials are running usually 1 to 4 years. They'll usually quit when everything dies. On average length of the trials are usually 2.5 to 3 year trials. And so these are total biomass for the trial. Okay. And so what we see here is the gains are actually not that great. And it reminds me of a trial where we did in 2007 where we were looking at gains, increases, and persistence. And what we see here is we are getting gains, but in newer varieties, there's vast differences between performance. But when we look here at these kind of benchmark varieties, we see greater gain. So I guess this is also, these would be gains in percent stand increases per year. And when we look at kind of benchmark or at specific programs, the gains are greater. So I wanted to kind of look at gains in my program. So I have 18 years of breeding. Our heritability on individual plant basis is 0.15. You're thinking that's not great. Actually, for individual plant selection, I'm pretty happy with that. Also, as part of this program, we can also do half-sib selection. So this here kind of shows our selection gains over time. The metric here is kind of a vigor score, but what we're seeing is a really nice gain of 1.9%. The key here for me is consistency. To keep breeding, you keep making progress. So, I mean, you just can't quit. Also, there's kind of a ramp-up time, until we get the thing going. This is base building, and then after a while, then you really start taking off. These are the top 10%, and this is just the average of everything we put out that year based on the year of creation. Now, I wanted to translate this into actually megagrams per hectare. A lot of people say, well, your space plant stuff doesn't represent your sward. Well, if you take this is against 160 sward trials, 123 space plant nurseries over decades. If we take, these are varieties that we have in 10 sward trials, and we have them as 80 individual plants in the nursery system. Well, guess what? Once you start have huge numbers, the correlations are real. So I'm actually thinking, in a variety trial, if you don't have 6 separate sites, possibly 10, most probably you're just looking at noise. And so really, to get accurate sward yield things, you really have to test it a lot. And what this shows me is our space plant system is really, it is effective at getting at yield, which we're trying to do. As a side note, fancy drones and stuff won't solve this problem. There's no substitute for having multiple trial sites. Just because you have more accurate measurements won't solve this issue. It's a G by E issue. So anyway, we can translate this now into our gains, and so our estimated gains per year is about 0.14 megagrams per hectare increase in biomass. So that's about, and this is just as you think, like, what's the potential? Where can we go? This is about the rate of gain that we can achieve using traditional breeding. So now I'm getting to the end. I have 2 slides. Yeah, so I just wanted to talk about other traits. I think Randy kind of, for me, these are the specialty traits, right? We have the big 3, persistence, seed yield, and biomass. But, the companies, they always want to have the specialty trait because they can market that. So these are some of the specialty traits that people are kind of looking at. I don't know if people are really making varieties. Like, there are people trying to make varieties with the stoloniferous rhizominous. There is some activity in the 2,4-D. There's talk about protein protection. I don't know if they've specifically made variety, and again, there have been varieties with lower phytoestrogen. So this is kind of my final thoughts. I tell the graduate students, this is the thinking of a breeder. So, this is the traditional breeder, and this is kind of like the university guy. So, as a breeder, I think you have to be kind of both. But what are our challenges? This is what I think about. Is breeding for these three enough? And which of the three is most important of those three? And, in some ways, sometimes I think, well, maybe I should just breed for seed yield because, if you talk to the company guys, they're like, well, yield and persistence doesn't market or sell. So, I don't, yeah. But these are kind of some of the challenges is how do we get, move forward with these other ones? Are our selection gain rates enough? And so I kind of showed you the number. What's even possible? Even if we had great new technology, how far could we accelerate it? And then being at the Dairy Research Center, I constantly talk to nutritionists. We're always competing against corn. I mean, in the end, your system, if we can't keep up with corn thinks they're going to do everything. So that's what I think about. So I think I'll leave it at that.
Let's thank Heathcliff. While I switch over, you can do the introduction.
While Derek is loading, I will give you just a little background on him and introduce him. Derek is Vice President for Forage Research and Development with DLF and the Grassland Manager, or the General Manager for Grassland Innovations. Met Derek in 1987 when he was working on his PhD at the University of Wisconsin. He's been 39 years as a clover breeder, pushing 40, he tells me, and in New Zealand with over 30 cultivars released in New Zealand, Australia, the U.S., and South America and Europe. He's current chair of the IGC Continuing Committee, and you've heard from him a couple of times on that. So,
Yeah.
Derek, thank you.
Thank you. You're probably already tired of that. My paper is titled Reimagining White Clover and its use in global systems. And I think actually Heathcliff's given a really good overview of some of the challenges that our forage legumes actually face. And if we want to make changes, we do have to tackle those traits. And I will show why we have to deliver seed yield, and it's not just so the company guys can sell seed. I want to reflect on Norm Taylor because when I was a young breeder, Norm was in his prime, or maybe towards the end of his prime, and he actually came and visited New Zealand before I came to the States to do my PhD, and he came to work with Margot Ford. And they delved through the Margot Ford, the germplasm centre that's now named for Margot Ford, and they took all the clover accessions, grew them out, characterised them, pointed out the ones that actually were in the wrong, they weren't the species that was labelled, and did a really good worker service. And then you can see some of the flower colours of some of those species which have already been covered. But a really inspiring individual. I will say though, however, he was possibly the slowest walker from the cafeteria to the glasshouse that I've ever seen, but probably because he was thinking so much. So in terms of white clover, it is still the predominant legume used in graze swards due to those features up there. Its nutritive value, its persistence under intense grazing, and its ability to fix biological nitrogen. Where its use declines is when synthetic nitrogen is cheap, where seasonal variation in rainfall or higher water deficits occur, affect the persistence, and then farmers lose confidence in it. It also declines a little bit where you get grazing tolerant red clovers that will do 4 or plus years in the sward, and they are very competitive. Also with grazing tolerant alfalfa in situations where they can manage that. Bloat remains a major concern for end users. Where use increases is when synthetic nitrogen is expensive. And then it becomes a much more efficient way of feeding the associated grass. Also, as we enter an increasingly regulated environment, it becomes more important because the end inputs are capped. Where we're seeing a major use, particularly in New Zealand, is where forage herbs, due to their high quality and grazability, have become a bigger part of those swards. And then white clover and red clover use have increased in association with those. And then lastly, the move with regenerative pastures, diverse pastures, has seen white clover almost always included in those, and it tends to be one of the components that dominates because of its persistence long term, whereas some of those other components do not always persist. In the paper, there's some examples of the economic value that white clover has added, and the ones there are a very small proportion of the studies that are out there. But Joe Boughton replacing the Ladino, short-lived Ladinos with a more persistent Durana, the estimate of that was added $86 US per hectare per year. In Ireland, the reintroduction of clovers into those swards and reducing the nitrogen inputs has increased the overall net profitability of those systems by approximately €300 per hectare. In New Zealand, Mike Dodd, I don't know if he's here, but he did a study that showed that carcass weight of cattle where white clover was introduced into hill country increased by over 30%, and the net profit of those systems increased by nearly 50%. Those are significant results and outcomes for the farmer, not for a seed company. And in New Zealand, the estimate of the economic value of white clover is over $2 billion a year, and those estimates vary, but that is year on year. And there is only 2 other species, plant species in New Zealand that rate higher than that. One's ryegrass and the other's pine trees. And then there's an order of magnitude drop below that. So it's an important species around the world. So how can we reimagine this? And this is the challenge for the next generation of breeders. My generation and the previous generation always leave more than enough for you to work on. There is always more to work on. And so I'm going to cover these topics and see if I can convince you that using the germplasm that Norm Taylor loved to work on and different breeding methods, added value traits for the end user, and improved seed yield, and that we also have to tackle environmental challenges. So in terms of using novel germplasm, we have over 30,000 accessions of white clover in the Margot Ford Germplasm Centre. We have made collections around the world, and almost invariably those collections have led to plants being included in cultivars that have been commercialised, and there are some examples up there. So if you take those collections and work with them, there is always novel genetics there to use. In addition to that, on the left-hand side of the slide there are 11 species related to white clover that have been crossed with white clover or through bridge crosses to bring genes in. All of those crosses have been made to this point with just random genotypes that would cross. And one of the things I believe that we could do in the future is to pre-screen those wild relatives for genes of interest for traits that add value in the future. In addition to that, we've got really good molecular diagnostic tools that I believe are adding knowledge for us to be able to look at the underlying genetics and genetic variation. I'll just go the other opportunity. If you look at the progenitors of white clover, which are pallescens, which was talked about earlier, Occidentale and pallescens, which were talked about earlier in the conference. These species are the progenitors. So white clover first originated about 45,000 years ago in the last ice age. There is the chance now to use those species to resynthesize white clover. What genes were narrowed through that glaciation that we have missed. What opportunities are there? As I said, the genetic relationships that underlie this really using those genetic tools are very powerful. Most of our commercial cultivars sit here, and we've got vast variation around that we can investigate and indeed are utilising to see where we take this crop in the future. Marker-assisted selection's been tried in itself, not really effectively because of the multigenic nature of most of the traits that we deal with. We're investigating genomic selection, and I'm not going to give you a lecture in genomic selection, but it utilises randomly spread thousands of SNPs across the genome to be able to try and then use that knowledge of the underlying genetics to get an estimate of the breeding worth of that material and indeed the kinship and relationships within that. There is the potential, and I do say potential because we haven't proved it yet, to reduce the breeding cycle. But it requires good prediction models and we've got the training populations going. We started the journey 7 years ago, and so we are now really at the point where we have to put up or shut up. The estimates from Grace's PhD suggest we can double the rate. Those trials are in the ground now to see whether we can, and whether indeed we'll probably go one more generation before we go yes or no. But there are risks with all of that. Population size and changes in your breeding methods are challenging. So I'm not going to say that's easy. It's something that we really have to tackle. But can we accelerate that rate of genetic gain? And I agree with Heathcliff that we can do that through conventional selection as well. So it's got to be better than that. To do that, we do need improved phenotyping tools. And I go, drones may not do it, but any phenotyping tools that allow us to increase the amount of material that we evaluate and the number of sites that we can handle for the same number of people helps me. We're working mixed swards with white clover all the time. All our breeding's done in that background. So to be able to assess clover content rapidly, to be able to look at stolon density will be important tools for us. I don't care what it is, but what the tool is, whether it's a drone or a LiDAR rolling over the ground, it has to be efficient and cost effective for us to use it. In terms of tackling environmental challenges, an inevitable outcome of animals grazing high protein content grass clover pastures is the formation of urine patches with higher end loading than the plants can utilise. And Glenn Judson, who's here at the conference, the average dairy cow is grazing 140 square metres of pasture every day, and they are then excreting approximately half of that nitrogen into 3 square metres of ground. So we need something to tackle that, and those of you who are here for the plantain session will have seen one possible tool. Our white clover pastures have lower soil carbon and nitrous oxide losses than crops. But we're not always compared against crops, certainly in countries where grazing is the primary use. Our nitrogen losses to groundwater are also lower than arable crops. Is that good enough? How can we enhance that? We can, by using clover more, whether it's red clover or white clover, reduce our reliance on synthetic nitrogen and lower the CO2 emissions associated with that fertiliser use. And I wonder if we can, as part of screening those wild relatives, look for those that do have natural traits that reduce methanogenesis and drought tolerance. Already talked about the last point. So in terms of how we might tackle the environmental traits, Condensed tannins have been the holy grail for all of our forage legumes for more than 60 years. And they increase the nitrogen use efficiency considerably. In this case, we believe around 60% in the rumen. If we can get condensed tannins expressed at that 1 to 3%, we not only reduce greenhouse gas emissions by 15 plus, 16 plus %, but we reduce end leaching, we reduce the deaths of animals to bloat, which is still a significant cost in all dairy areas where there is grazing. And at the same time, we get the benefits of animal productivity gains of the efficiencies of condensed tannins. So we are working on this, and the top slide there is the naturally occurring condensed tannins that already exist in white clover and in red clover in the flowers. So they're there, but they're not expressed in the leaves normally. What we've done is created, these are transgenic, but they express condensed tannins in the lower epidermal layer in the leaf, and indeed when we do the in vitro tests we get the reductions in methane that we expect based on all the animal work that has been, animal nutrition work that's been done with condensed tannins. It works, it's effective, it's transgenic. So in terms of what we've achieved to date, and I've got 5 minutes, you can read these, but we've got stable expression across generations of 2 to 4%. We've demonstrated the reduction in methane, and we've actually field tested in the US, and we're now currently field testing in Australia. I said I'd get to seed production, Heathcliff. This is a comment from one of our leading arable growers in New Zealand. And I think it cuts to the core of why seed production is important, is that for our forage legumes, we compete with every other crop out there for the economics of seed production. There is no free lunch. We have to be competitive with other crops. We can in white clover. There's a couple of published, well, one published, one unpublished example of where we've done this in white clover. We published on the work in 6 commercialised cultivars which had around an 800 kilogram per hectare yield. The final products that went to market were consistently 20 to 30% better for seed production. We've just done the same with a lower producing European cultivar, and that's the increase through one cycle of selection is around 40%. Not difficult, easy to achieve. We've got to make sure when we do that, though, we don't lose the underlying agronomics of those cultivars. So, in summary, for me, reimagining white clover will include improving our breeding methods, capturing traits from wild relatives, improving persistence undergrazing, which I didn't cover really much in this talk, delivering traits that add value, not for a seed company, but the end user farmer. Because that's where you generate value, is if we can demonstrate and capture value on farm. And I believe we must have and must continue to look for traits that improve the economic benefits that our farmers need. So I think I've got 2 minutes for questions.
You've got about 2 minutes, so questions for Derek, please. I'm sure somebody has a thought on your mind. In the back, speak up.
When are you going to have a drought-tolerant white clover? I understand Vickers.
You're saying you want a drought-tolerant white clover, Laurie?
Yes.
Yeah. Yeah, so that's a... Do you want drought or do you want phosphate? I'll answer the drought question first. We have got cultivars already that have demonstrated better drought tolerance, and they've been sold in New Zealand. We basically used harsh environments in Australia to do that. Is that drought tolerance enough to tackle our changing climate? No, it's not. But you've got to look within the limits of the species. So that's where the wild relatives do really come in and play a part, Laurie. Phosphate use efficiency, a totally different and much more challenging target.
Just step this way just a little bit and let Phil go ahead and load and
Yeah,
take
sorry.
one more question.
Down the
Another question. Dennis.
I'm really excited to see the outcome of the condensed tannin white clover work. Excited to hear that some of those yields, forage yields, are increasing and are going to come. Could you comment a little bit about where you see that fitting in? I know it's especially important in New Zealand because of the requirements of methane reduction in your country as well as European countries as well. Comment about that?
I think the trait fits everywhere where clovers are used. I think the regulatory environment is the challenge. So where can you, it is transgenic. I don't know, 50, 60 years of work, we haven't tackled it any other way. So it's then a public debate around the value of the technology for society and for end users because of the multiple benefits. Australia, there's a clear pathway to market, regulatory pathway to market. US, there is, but it would be challenging. Some of the other, South America normally follows North America. Europe will be almost impossible.
You're introduced?
Oh, I am.
Now I'm battling with the technology here.
Yeah. All right. The next speaker is Professor Professor Phil Nichols from the University of Western Australia. Phil has worked in annual legumes for approximately 38 years. He now works on a joint venture, Alba joint venture, to develop the next generation of these and also to train and equip the next generation of breeders. So with that, I'll hand over to you, Phil.
Thank you, Derek, and thank you to the organisers for inviting me. I was given the challenge of talking about annual clovers around the world.
So that's the other 14 species that we haven't talked about so far, right?
So I thought, how the hell am I going to do that? I know my patch. I don't know much about, I know in some cases some. So what I did was I recruited a range of clover experts from the main regions where annual legumes are grown. So I'd like to first of all thank all my co-authors for their inputs. Okay.
There you go.
Rightio. It's not showing all the pictures up. Why would that be? Interesting. I've got lots of lovely pictures here, and I want to follow on from Ken's lovely talk to show the diversity of all the annual clovers, their beauty. It's a magnificent genus to work with. So, okay, it's not quite showing all the things I had. Let me just have a look here. I'll use this one then.
Right button on the pointer.
No, it's okay. Yeah, it's not showing all the pictures. Never mind. It's not showing the pictures. It's not going to show all the beauty of the clovers. But anyway, we'll do our best here. Okay. As has already been discussed, the importance of annual clovers, firstly, as they're a nutritious livestock feed, both for in situ grazing and also as conserved fodder. And of course, the other important thing they do is they convert atmospheric nitrogen into forms that can be utilised by plants, and that's of course is in symbiosis with the appropriate rhizobium strains. And so that nitrogen can be utilised for the growth of the clover, and it's also available for other non-leguminous pasture plants, herbs and grasses that are in the pasture, and that nitrogen is also available for utilisation by subsequent crops. And that's one difference with many of the annual legumes. They're often used, certainly in our country, in rotation with crops. As Ken has pointed out, there's about 200 on the basis of these two seminal publications. there's about 255 recorded trifolium species, and 155 of these have an annual life cycle, with another 3 that have either annual or perennial forms. So where do these come from? And Ken has touched on some of this. Well, roughly 2/3 of all the annual legumes originate from the Mediterranean basin and surround. And interestingly, it's the origin for all 14 of the annual legume species that have been commercialised. There's another 14.6% from sub-Saharan Africa, nearly 18% that are unique to the USA, and there's another 4 from Chile. So what are some of the key adaptive features of the annual legumes? Well, firstly, they're adapted to winter rainfall dominant summer dry areas. So we're talking about particularly Mediterranean type climates, or those that are temperate climates that are more summer dry. They're long day plants. They germinate in the autumn winter. They grow through the winter spring, and they senesce in late spring or summer after they've set their seeds. So the time to first flowering is genetically controlled. It's modified by latitude, altitude, and cool season temperatures. So in low rainfall, short growing season areas, early flowering types are adapted so they can set seed early before the onset of the summer dry period. But in high rainfall, long growing season areas, late flowering types are more suited, because they can take advantage of the long growing season and still have time to set seed before the summer drought. And one of the other key features of the annual clovers is that many of them are able to self-regenerate from seeds set in previous seasons, and so this leads to persistence in native grasslands and also sown pastures. Okay, so I'm missing all my pictures, which is a real shame. I know, I'll have to go plunder on. My apologies, but anyway. So one of the things about annual legumes is that many of them are hard seeded at maturity, at seed ripening, and over the summer period, many of these become permeable or so-called soften in response to warm summer temperatures. And these are available for germination in subsequent years. And the remainder of these that aren't ready for germinate, they form a seed bank of hard seeds that are available for germination in future years, and allows regeneration after seasons of little or no seed set. So this character is genetically controlled, and it varies considerably between and within species, and allows for selection for lower or hard seeded types. So generally in unreliable rainfall or short growing season areas, we need higher levels, or plants with higher levels of hard seedness tend to be the best suited, but in more reliable, longer growing season areas, lower levels of hard seedness are required, because you need a bit of insurance in case there's some dodgy seasons or not. But importantly, for annual fodder types that are resown each year, we don't need hard seeds. Okay, so I've listed here 14 annual clover species that have been commercialised, and within those we've got 2 varieties of Persian clover, botanical varieties, and 3 subspecies of subterranean clover. So we're talking about 17 botanical taxa that have been commercialised. Okay, so how do we look at the importance of the different annual legumes? One way is to look at seed figures, and we've got quite good figures of certified seed production in Australia, and this shows you the mean over the last 10 years of tonnes of seed sold. And here we have subterranean clover, which is the dominant species for certified seed production, with Persian balança quite important, and a bit less so with arrowleaf persimmon, and these are the other ones. But that's all very well, because seed production figures are only for certified seed. And much annual legume seed is produced and sold as uncertified seed. There's a lot of over-the-fence trading that isn't captured, so really the total seed production is unknown. And the seed production figures for other countries, importantly, in this case, it's difficult to obtain. So what we did was to overcome this, we developed a survey, basically, of where the co-authors gave a 1 to 5 rating for the importance of the annual clovers in their farming systems. And so it's a qualitative measure where 1 is rarely sown, 3 is moderately important, and 5 is highly important. So this just gives you a very quick summary, but it shows you the regions being Southern Australia, New Zealand, Southern USA, centred particularly around the Texas region, Southeastern USA, around the Florida region, Western USA, around the Oregon and California, Southern Europe, Mediterranean regions, North Africa, West Asia, South Africa and South America, predominantly Chile. So I'm going to talk a little bit more about these species in particular. No pictures, never mind. So the most important globally is subterranean clover. Its main feature is that it's highly grazing tolerant due to its prostrate habit, and Ken put up a lovely slide showing its burr burial ability, and this protects the seeds from grazing over the summer months. So there are 3 subspecies: subterranean, which is adapted to well-drained, moderately acidic soils. Then we have the Ininicum subspecies, which is adapted to poorly drained soils, so it's used quite commonly in wetter areas. And subspecies Brachycalycina, which is adapted to more neutral to alkaline soils. And it has poorer burial than the other 2. And that data's missing too, but never mind. There we go. You'll see here that this is important across all these regions, mostly. And the subterranean type is more important than the other 2. So Balansa clover is mainly grown as a self-regenerating pasture, certainly in Australia. It can be used for grazing and cut for fodder. It's got high waterlogging tolerance and mild salinity tolerance, so it's commonly grown on soils prone to waterlogging, particularly if there's a little bit of salinity around. It's an important species. It's small-seeded but has vigorous spring growth, and it's important in Australia and South Africa, moderately important in Western USA, and less important in some of the other regions. And there's breeding programs in Australia and the Western USA, particularly in the Oregon regions. Persian clover, well, there's 2 botanical varieties. The major variety is very erect, large leaves, thick hollow stems, and it's generally grown as a 1-year fodder crop. It's traditionally been cultivated in Iran, Afghanistan, Pakistan and the Middle East. It's sometimes sown in Australia as a high quality forage, occasionally in South Africa and in other regions. And there are breeding programs in Australia. Variety Rasupanana is more of a pasture type. It's a dual purpose, or it's a self-regenerating grazing and fodder species. It's more hard-seeded. It's not as erect as the majors types. And there's some breeding programs in southern Europe. So it's got some importance in particularly in Europe, southern Europe, and some importance in southern Australia. Okay, crimson clover. I had some lovely pictures of crimson clover, but you've seen some already from Ken. It is one of the most magnificent clovers. It's a erect habit with crimson flowers. It's used for grazing early on and can be cut for very good quality fodder. It's the most important of the annual clovers in the US. So it's widely sown in southern and southeastern USA, primarily to increase the winter biomass of warm season perennial grass pastures, and it's generally sown and it's also sown as a 1-year fodder crop in southern and central Europe and western USA. We don't use it very much in Australia at the moment, but there may be opportunities to do so. and their breeding programs in the USA and Europe. Arrowleaf clover, Trifolium vesiculosum. It's got an erect habit, very erect habit. It's slow growing early on, but once it gets going, it really powers away. You can graze it early on and it makes good fodder. It's quite hard seeded and in many environments will be self-regenerating. It's later flowering and more deep rooted than most of the other annuals, so it means you can get green feed later into summer than other annual legumes. So it's commonly used in southern and southeastern USA to increase winter biomass, again in these perennial grass pastures. It's sometimes sown in southern Australia, the western USA and Chile, and less so in Australia, in Europe and New Zealand. And there's breeding programs for it in Australia and the USA.
5 minutes.
Okay, Berseem clover is primarily grown for fodder in irrigated or high rainfall pastures. It's best adapted to fertile, medium to heavy textured soils. As the name suggests, Egyptian clover, it's commonly used in North Africa and also West Asia in cut and carry systems for dairy and beef cattle. It's often grown also in southern Europe and Chile, and occasional use in some other areas. And there's breeding programs in the US for it. So other less widely sown clovers, squirose clover or Trifolium squirosum, it's often sown as a one-year fodder crop in southern Europe. Ball clover or Trifolium nigrescens, sometimes sown in southern and southeastern USA. Rose clover, it's a relatively minor reseeding pasture species in western USA, southern USA and southern Australia. Cupped clover was developed in the 1960s as a low rainfall southern Australian pastures, but it's rarely used and seeds difficult to obtain. Purple clover is a deep-rooted dual-purpose alternative to arrowleaf clover, but it's rarely sown in Australia and not really used anywhere else. Okay, so there's been some recent domestication of some new clover species since the late 1990s, and the key issue here was subterranean clover is very widely used in Australia, but it's difficult to harvest the seeds. So new species have been developed that you can harvest seed directly with headers. So three key ones, or three of them, have been bladder clover, Trifolium spumosum, which is a prostrate habit and aerial seeding, and there's some breeding work occurring in Australia. Gland clover has got really good resistance to red-legged earth mites, which is a major problem in Australia, and to aphids. It's got quite low biomass potential, but is useful in mixed pastures. And then eastern star clover, Trifolium dasyurum, was released a few years ago, and its main feature was it's got very delayed softening of hard seeds, and it allows weed control prior to the regeneration in the second year. But seed here is really rarely available. So bladder and gland clovers are sometimes used in Australia, and there's a little bit of use in southern Europe. There's a range of endemic and naturalised clover. I had a lovely panel with lots of ones, but Ken has already shown many of these. But they're important in many situations, as even though they're not sown, they do contribute to annual production, either in the native grasslands or also as naturalised species in Australian parslands. So I've just listed some of those there, and these are just a subset of the ones that are important. So I'll just finalise with the future prospects for annual clovers. So the areas sown to annual clovers may well increase in future years. Some of these have already been mentioned. Nitrogen fertiliser costs for crops and grass-based pastures are becoming more prohibitive cost-wise. And in some areas there's increasing government regulations to reduce infiltration into river systems and groundwater, and annual legumes can supply nitrogen in these situations. Climate change will and a more erratic climate. We're already seeing evidence of that. So we'll see some areas become drier and more marginal for some of the perennial clovers over summer. So late flowering annual clovers can have a fit into those. So the challenge here is firstly for plant breeders to develop better adapted pasture legumes, and to agronomists to develop improved farming practices. So there's a likely role also for new clover species with important adaptive traits, but there's a difficulty in developing the markets for new species that would need to be overcome. The Forage Plant Genetic Resource Centres, and of course Norm Taylor and the Margot Ford Centre, and the one we have in Australia, and there are other ones as well around the world, they'll become even more crucial, I believe, as to identify plants with the characteristics that we need for future resilience. And finally, biotechnology is likely to become more important in breeding programs, at least for some of the major annual clover species. Firstly, for the provision of new genes for important traits, and also genomic screening tools to allow us to do selection from multiple traits. So I'll finish there, and I had all the photos of all the co-authors, which I'd like to thank very much for all their inputs. So
Thank Phil for that presentation.
all right.
Unfortunately, we don't have time for any questions, so Joe, we're going to move straight to you. Thank you. Thanks, Phil.
Okay.
Close that one and we should. There you go. So our final speaker is Dr. Joe Boughton. Joe says he grew up on a family farm in the Mississippi Delta. Undergraduate degrees at Mississippi State, graduate degrees at an institution that I'm pretty familiar with, the University of Florida, which is where I first crossed paths with Joe about 1975, I think. He went to the University of Georgia, progressed through the academic ranks for about 30 years, was for about 10 years a director of research at the Noble Foundation. His entire career has been involved with research on forage breeding with numerous imported cultivars produced, and he's currently a consultant for both private and public institutions. Thank you, Joe. Please give us your talk.
Thank you, Ken. One time somebody asked my daughter, what do you do? She was 6 years old at the time, said, what does your daddy do? And she said, well, he's a doctor, but he can't help you. So that's a lesson to a lot of young people out there. If you start feeling proud, a little more proud of yourself, tell a 6-year-old what you do and then have them introduce you. And then you'll see where you stand. I really like quotes. In fact, there's a book that I was one of the co-authors, a minor co-author with Don Ball and Gary Lacefield and Carl Hovland and Vivian Allen downstairs in the forages section on quotes. And I like to use a lot of quotes, and I'll use this one. I thought this was a great one from Emily Dickinson. She was a poet, and I thought she nailed it right here in very few words. To make a prairie, it takes a clover and one bee. And I think on reseeding, as Phil pointed out, there's a lot of reseeding clovers. Another quote from Mark Twain, can't get out here, said, prediction is difficult, particularly when it involves the future. And that's what our talk is, John Caritas and I. So, and another quote I haven't got up here was from a guy named Samuelson. He was a big economist, and he said, people who predict the future fall into two groups. Those who don't know, and those who don't know they don't know. And John and I have, just by the fact we're going to proceed with this talk, we believe we're in the second group. We just don't know we don't know, but we're going to say something. And Ken, I really got confused here. I got 577 species. I got that off the, I must have just read it, I got it from the Nordic Baltic thing, but anyhow, there's hundreds. And the main thing for this talk, for Norman Taylor, was that Norman Taylor collected, I got this one collection he reported in the Southern Pasture meeting, where he and Dick Smith and Bob Langford had gone into Europe And they had collected 37 species and 291 successions that's in the collection there. So Norm, again, was very impactful collector, as well as a breeder and a geneticist of clovers. Now, seed sales are, as many of you know, are limited to a very few species, but the cultivar numbers are high in some species, like white clover and red clover. And also, of course, the main reason we use clovers is to mix them with grasses, in pastures especially, for better animal feed, increased seasonal forage production, and, of course, biologically fixed nitrogen. That's been talked about a lot of times, but those are the main important things. The uses, current uses for farm income and ecosystem services, everything from pastures, again, and mixed pastures, either bicultures, especially a grass and a legume together, but definitely in New Zealand, of course, they talked about perennial ryegrass and white clover. In the United States, you really can't beat tall fescue and white clover. It's very economical. And then, of course, you have hay production. Here's, we're really into some cover crops now, use them as cover crops and rotations. We use a lot of them, we have a lot of wildlife mixes, and a lot of clovers go into wildlife mixes along with grasses. And even in some of the there's a movement too for polyculture pastures. Seed production, this picture was of course taken in New Zealand. You probably recognize some of those people, Derek. And then, erosion control. Here's one that the Californians were popularizing in the 1980s. So, as a breeder, we're going to take a lot of credit here. We're going to pat ourselves on the back and almost break our arms doing that, but I'm going to tell you that really there's been a lot of successes with just straight selection, recurrent selection for half-performing families in grass and undergrazing. Put them in those grasses, graze them, look for the best ones, especially for persistence, because persistence is still the key. And also, as Derek mentioned, intergressing genes from related and wild species have been very important in the past and will continue in the future. And then enhancing the interaction with beneficial microbes, we're really talking about rhizobium. Yeah. So let's just real quickly, when you put white clover, in this case, we had a trial where we put it with that E+ there, that means it's toxic. It's a toxic fescue, tall fescue. This shows things as clear as any data I've ever collected myself. And I like to use this term competitive edge. It gives it a competitive edge. I picked that off a New Zealand publication. and that is, but there's something magical happens when you put clovers with grasses and graze them with animals. And this shows that there's also shows a couple of things going on. This was no nitrogen. This was, from the spring treatment, we put on 65 kilos of nitrogen per hectare, and then and again in the autumn, we put on another 65 kilos. So, but no difference in yield. So I like to use the term nitrogen replacement value. What are we replacing here? We're replacing 130 kilos of nitrogen to get the same yield. And that yield is a lot better when the clover, you can see that yield translated into animal gains. So that increasing yield from clover replaced that nitrogen and also gave us that competitive edge in boost in production of those animals. When we look at profitability, this was another study done by John Biermacher and his group, and this work I think was done in collaboration with the University of Arkansas, but again shows many of the same things, but now we're looking at it from an economic standpoint. And again, a toxic fescue, we were losing, if you're using toxic fescue, you're losing money. I mean, literally, you're losing money. I don't even know how they do it, but they do. But anyhow, they usually supplement is what they do. But you just go from here to here, a toxic fescue with white clover, and at least you're starting to make money, not a lot. You start moving into the non-toxic fescues, and it jumps up again. And then even though you're not dealing with toxicity, there's that bump again. There's that magic. Boom, animal performance goes up. Now, what about cover crops? Phil, I stole this off the Western Australia website, but these lay farming systems, I've always looked at them as they're the gold standard. These Australian farming systems, rotations, these lay systems where they're rotating, especially with subterranean clover and coming with an annual crop, wheat in this case, has been very remarkable in its abilities. Now, in the United States, we're starting to look a little bit more at using legumes in two ways. And of course, I'm sure it's been mentioned a couple times. If you're not talking about corn, you're not talking about anything, right, Heathcliff? I mean, it's all about corn. We're always trying to help the corn people. And they never try to help us, but we try to help them. But we're looking at using them in two operations. The annual legumes, the annual clovers especially. This is of course, as Phil mentioned, this is the crimson clover that we use a lot in the southern United States. And then we cover the crops. We look at we use that as a cover crop. Then we come back and look at growing corn, especially maybe even soybeans or cotton in our case, with very limited nitrogen, and seeing about what's happening to the ecosystem, what's happening to the yields, and so forth. We've also been looking at a little bit as using them as what's called a perennial mulch, a living mulch, where you use a perennial clover. In this case, ambiguum, or we call cura clover. Now, what has been the problem? Well, the problem still remains persistence in these type of environments. You put them in grasses, you graze them, you go through harsh environments, especially with droughts and stuff. You've got to be able to compete. The grasses are going to take care of themselves, but the clover, you've got to make sure that you keep a good percentage of clover in that pasture. If you're looking at the annual clovers, one of the big problems has been that even though we document every time we have a clover and even cover crops of all types, but definitely grass, clover, or whatever the cover crop is, you're really going to increase the what's called ecosystem services, soil environments especially. So that's always beneficial. But production and economic benefits have not been so consistent. Sometimes the following crop yields with corn, soybeans, sometimes they're down. And sometimes we need to look at net economic return, if you can use that for some grazing. But anyhow, and then also the clovers in this case, they just don't get going, and they don't, when compared to the grasses. So that's another reason they probably use them a lot together, but they need to be more competitive. The perennial clovers have the same problem as a living mulch. A lot of soil health, however you define it, has been improved. In this case, we were using Durana clover, and then there was a follow-up corn yield was about 10% reduced, and but maybe net economic return, they didn't get a lot on net economic return. So there's an inconsistency of results. Then we're talking about carbon farming, and in the U.S. we're still about corn and or soybeans, and so all the cover crop stuff fits into this carbon farming, and it's mainly using an off-season and annual cover crop system that includes clovers. But more research and farmer experience is going to be needed consistently do this. You must have a registry now, and government's not running that registry in the United States. So what that registry does is they find development partners who can aggregate all this stuff. I'm not familiar with it myself. I've just read about it and heard of some seminars on it, but the current problems for cover crops using Clovers cover crops are going to carry over into these carbon farming systems. And so that's again where it's going to have to be looked at a lot closer. And as you can imagine, if you're going to sequester carbon, you better have good validation, with soil sampling, et cetera. I thought that was pretty interesting this morning that Sarah Place said, but maybe it'll even be easier to document methane, where you can get get credits for methane as easy as you can for soil carbon. So future, here's our future. We're going to have different systems research. There's no doubt about it. We've got to increase the use, productivity, and persistence and economics of these new clover varieties. And they're going to have to be within sustainability and carbon mitigation strategies. It won't be for me. I can do precious little at my age, but for those that are all in the room, that's probably going to have to happen. Seed coating, still important. Rhizobium still going to be important, maybe even more important going forward. So more work still needs to be done there. How to mitigate planting costs and revenue losses. If you're going to use these things, you can't be losing money. I mean, that's been one of the problems. And finally, you're going to have to do several years of work in your target geographies, I think. Breeding better varieties, Derek mentioned some of it. John Curtis, my co-author, had another paper in 2021, several authors on that paper, but he's predicting, and those authors predicting, that conventional breeding in all clovers and genetic modification in some species are still going to be important and lead to additional benefits, especially for pasture resilience and environmental outcomes. And again, probably going to look at genome mapping. Derek mentioned it. I won't go too much in there. Maybe genome by sequencing, the GBS approach, and using them to structure populations. I think that's always been an overlooked thing with the genomics. Some of them are affordable. Derek mentioned the CT using getting condensed tannins produced in some of these clovers. That's going to be very important. But that's again a transgenic approach. So you're going to enter the Alice in Wonderland world of regulatory. I've been there, done that, and I got a lot of scars on my back, and I don't plan to do it again. But it'll have to be done. And he mentioned improving seed yield. It's got to be cost effective, especially for any species. Early in my career, I was working on alfalfa those days, and I met an alfalfa grower in the West, and I was telling him I was producing some varieties for the Southeast, and he told me, he just looked me right in the eye and said, "Son, if your variety doesn't produce one more kilo," he was saying pounds, one more pound of seed than the one I'm growing, I'm not going to grow it. 1 pound. I said, all right, I'm going to go for seed yield too, so that you better go after it, yeah. And John Curtis, again, in his keynote address in 2021 IGC, talked about if you're going to go into these, especially genetic modifications, it's really going to challenge us, for integrating a large number of agri-tech developments into these current pastoral systems without negatively impacting sustainability. That's going to be a real challenge for you people. I mean, it's not going to be me. I can do precious little, but as we go forward, that's going to be tough. So I like this picture. I took it, so I know if it's a rising sun or a setting sun. What do you think it is? How many think it's a rising sun? Setting sun. It's a rising sun. So the future's bright. But I think clovers will continue to be extremely important. We're going to continue to look at them. I think they could become more important as we go forward in these new sustainability, carbon mitigation strategies, carbon farming as we go forward. And as Derek mentioned, if we can ever get enough condensed tannins, we might even look at the CO2 level in methane. But getting this thing is more dependable, and having a better economic return on their input is going to be important also. And future research must find ways to overcome this imbalance, I think. So anyhow, I thank you, Derek and Ken, for inviting me to give a talk. Yep.
Thanks, Joe. We do have time for a couple of questions. And if somebody can now hit the lights at the back, we're allowed to, or the front, we can turn them on again. That's better. That's great. Thank you.
Yeah.
Any questions for Joe?
Yes, sir.
I see you put some Kentucky clover on that intro.
Yeah, I meant to point that out. Sorry about that.
I actually grew the plants. I was really
Yeah, I should have pointed it out. Sorry.
With that in mind, I mean, how much of the future of Clovers is about breeding improvements on existing plant material, or are we going to be doing some more discovery in some of these natives, especially here in North America?
Well, as I mentioned, we've been successful in the past by dragging in traits from related species, and that's where this becomes very important. I remember reading a paper from the 1981 IGC by Jack Harlan, and he talked about curation of these collections, and he said, but the breeders make crosses, and when in doubt. So rule 1 is make crosses. Rule 2 is when in doubt, refer to rule 1. You know, just keep making crosses, and you're probably going to release some stuff and unleash some stuff that's been tied up in there. So again, I think the collections to be curated. It's always been important to curate collections and to just have them there for the breeders to use whenever they can.
Is there another question? Because I think that was actually probably the most salient comment of the whole 6 papers.
Thank God.
Make crosses.
My daughter would be proud.
Right, if there are no more questions, we'll call the session to close. But I do just want to thank Ken for coming up with the idea for this seminar to honour somebody who was an iconic Kentuckian, an iconic clover researcher, and actually no longer a living legend, but certainly a legend. And I want to thank each of the speakers, and if you can join me in thanking the speakers for making this a really interesting and enjoyable session. Thank you.